Welcome to the deep dive. This is where we sift through the scientific noise, take the most complex research, and distill the exact insights you need to be fluent in the molecular future.
Today we're focused on what is, well, one of the most transformative technologies of the 21st century, CRISPR-Cas genome engineering. We're looking at a remarkable, really accelerated history. It only spans just over a decade, but it has fundamentally altered molecular biology.
And our sources for this deep dive really reflect that speed, don't they?
Uh-huh.
We're pulling from the earliest foundational papers, the ones that first dropped a working version of Cas9 into human cells back in 2013, all the way up to clinical trial results that were just announced in 2025.
Exactly. It's an incredible timeline.
That rapid evolution is genuinely astonishing. So today we are going to trace the path that took this technology from what was, essentially a blunt molecular hacking tool, a pair of imprecise scissors, really, to what we now refer to as a highly specialized, programmable, precision genome word processor.
That analogy is perfect, and it really sets our mission clearly. Our goal today is to synthesize that systematic engineering process. We need to understand how researchers took this original bacterial system, identified its flaws, and then just systematically engineered new Cas variants, fusion proteins, and delivery methods.
All to overcome those two big initial limitations, right?
Right. It was all about dramatically enhancing safety and specificity, while at the same time broadening the entire spectrum of genetic edits you could possibly make.
Okay, so let's unpack this by starting at the very beginning, the foundational concept. Before it was a lab tool, it was an immune system, which is a detail that I think is really easy to forget.
Absolutely. CRISPR, which, stands for Clustered Regularly Interspaced Palindromic Repeats, and the CRISPR-associated protein, Cas, they form the adaptive immune system of prokaryotes.
Like bacteria and archaea.
Exactly. So when a bacterium gets invaded by something foreign, like a virus, it actually incorporates these tiny segments of that foreign DNA into its own CRISPR array.
It's creating a kind of genomic mugshot file, a memory.
It's a lasting genomic memory. That's a perfect way to put it.
So then when that same virus shows up again, how does the bacterium use that memory segment?
So that memory segment gets transcribed into an RNA sequence.
Right.
Now, in the version we use most often, the one from Streptococcus pyogenes, the native system actually uses two RNA molecules.
Okay.
There's the CRISPR RNA, the 2R RNA, which is the actual segment of memory, the mugshot. And then there's the transactivating cRNA, or tricRNA, which basically acts as a scaffold.
And in the lab, we just fuse those two together,
Yep.
right? To simplify things.
We do. We fuse them into one synthetic molecule, the single guide RNA, or sgRNA. It just makes the whole system much easier to program.
So that sgRNA is essentially the zip code. It directs the Cas9 protein to the exact location on the target genomic DNA.
Through Watson-Crick base pairing, yeah.
But what locks the system onto the site? What makes sure that the cleavage only happens at the exact right place? There has to be a confirmation signal.
There is, and that's the protospacer adjacent motif, or PAM. The sgRNA gets the Cas9 protein close, but the Cas9 needs that short conserved sequence immediately adjacent to the target site to really lock in and confirm its binding.
So for the common S. pyogenes Cas9, that PAM is 5'-NG3R.
Typically, yes, and this is a critical point. No PAM, no cut. The target sequence has to be there, and it has to be immediately followed by that correct PAM sequence. It's a two-factor authentication system.
Okay, so once that entire complex is long gone, you've got the sgRNA, the Cas9, the PAM, what was the fundamental immediate outcome of that wild-type Cas9 activity?
Cleavage. The Cas9 enzyme uses its two nuclease domains, H and H and RuVC, to generate a double-strand break, or a DSB.
Right through the DNA.
Right through it, typically located 3 base pairs upstream of that PAM sequence. And that DSB, well, that's the source of all the initial excitement and, critically, the source of all the initial problems.
And here is where we hit that first massive challenge, the one that really limited CRISPR's therapeutic potential in its early years, off-target effects. Even with a 20-nucleotide guide sequence, which sounds pretty specific, why was the wild-type Cas9 so prone to making mistakes?
Because the binding just wasn't as strict as we initially hoped it would be. Studies very quickly showed that wild-type Cas9 could tolerate multiple mismatches between the guide RNA and the target DNA sequence.
And this tolerance wasn't just random, was it?
No, not at all. It depended heavily on the number of mismatches, their precise position along that guide sequence, and even the specific base identity of the mismatch. Generally, mismatches closer to the PAM sequence were tolerated less, but the tolerance was still, significant enough to be a problem.
So if that enzyme binds to an unintended site that's only partially complementary, you get an unwanted off-target double-strand break.
Yeah.
And that leads to a whole cascade of problems once the cell tries to fix it.
Precisely. The cell's default high-speed repair mechanism for DSBs is something called non-homologous end joining, or NHEJ.
Which is the fast and messy one.
The fastest and by far the most error-prone. It simply glues the broken ends back together, and in doing so, it often results in these semi-random insertion or deletion mutations we call indels.
And if those indels happen at an off-target site, say inside a critical tumor suppressor gene, that's a catastrophic safety risk.
It is. It's a huge risk. And not only is the outcome unpredictable, which is terrible for any kind of precision editing, but the very act of inducing a DSB, especially over and over, is incredibly stressful for the cell.
It can trigger the cell's own safety mechanisms.
Exactly, like p53-mediated apoptosis, which is just programmed cell death. So for any in vivo application where you're trying to edit millions or billions of cells inside a living patient, this lack of specificity and the inherent messiness of that repair pathway were just severe, severe limitations.
So moving from a fantastic research tool to a safe, reliable therapeutic required solving this specificity challenge right away.
Immediately. It was priority number one.
That provides the perfect transition into the first major phase of engineering, enhancing fidelity. The goal was clearly to reduce those unintended bindings. So how did the earliest rational design efforts improve Cas9's intrinsic ability to tell the difference between a perfect match and a site with a minor mismatch?
The initial strategy was purely biochemical. Researchers started introducing very specific mutations into certain amino acid residues of the Cas9 protein itself.
Targeting the parts involved in DNA binding and cleavage.
Exactly. The goal is to create these new variants like EC-Cas9s, pCas9, HF1, and SuperFi-Cas9.
And how did these mutations actually translate into higher fidelity? Did they just stop the Cas9 from binding so weakly in the first place?
Not exactly. They installed what you could call a kinetic proofreading mechanism.
Okay, what does that mean?
Well, think of the wild-type Cas9 like a simple spring-loaded trap. It binds and it cuts. It's very direct. These high-fidelity variants are more like a complex multi-stage lock. The mutations actually prevent the enzyme from properly completing all the necessary conformational changes when it's bound to a mismatched target.
So if the enzyme encounters a mismatch, it might bind for a second, but then it gets trapped in an inactive state. It's physically unable to complete the structural rearrangement it needs to actually engage the nucleus domains and trigger the cut.
That's it, exactly. For instance, the SPCas9 HF1 variant showed a really dramatic reduction in off-target editing. It maintained high on-target activity, but just dropped the off-target events way down.
So this was a critical first step because it addressed the non-specific binding problem head-on.
It was, but it didn't entirely eliminate the risk, and it didn't change the fact that when it did cut, the outcome was still subject to that messy NHEJ repair.
That sets up the next major mechanical innovation, one that tackles the problem not by making the enzyme smarter, but by making the required sequence of events much more complex. That's the Cas9 nickase, or Cas9N.
The nickase was a brilliant simplification. Instead of relying on all these subtle structural changes to stop the cut, researchers just physically disabled one of the two nuclease domains.
The D10A or the H840A domain.
Right. They just broke one of them. And this meant the resulting enzyme could only generate a single-strand break. A nick, not the catastrophic DSB.
A single nick is relatively harmless, isn't it? The cell's repair machinery can fix that pretty easily.
Very easily, with very little error. But to leverage this weak nick to actually get the editing outcome you have to pair it. And that's the dual nickase approach.
That's the key. The dual nickase approach uses a single Cas9 enzyme and two separate guide RNAs.
Correct. And they target adjacent opposite strands of the DNA. The guides are spaced by a specific offset, which can range from, say, 20-something base pairs, like the 23 base pair offset they used in the early EMX1 testing, up to maybe 64 base pairs in other studies.
So the genius here is that to generate a functional DSB, the event that actually triggers the repair pathways, you don't just need one perfect match. You need the precise coincidence of two separate high-fidelity binding and nicking events happening close enough together.
Precisely. If one of your guides binds to an off-target site by mistake, what happens?
You just get a harmless single nick, which the cell cleans up.
Exactly. But if both guides bind to their respective intended opposite strand targets, the two nicks are close enough together that they destabilize the DNA helix, and you get what looks like a clean DSB.
So the specificity requirement is essentially multiplied. The odds of two separate off-target events happening at the same time in the same place are astronomically low.
It's multiplicative. If the chance of an off-target event at site A is 1 in 100,000, and the chance at site B is 1 in 100,000, the chance of both happening simultaneously in the same region is, well, 1 in 10 billion.
That multiplicative specificity must have resulted in some truly impressive data compared to the wild-type Cas9.
It really did. The early evidence in human HEK 293FT cells was definitive. They tested it on the EMX1 and VGFA genes loci that were known to have significant off-target problems with wild-type Cas9.
And the results?
The double nicking approach achieved editing efficiencies that were comparable to wild-type Cas9 on target, but crucially, it showed no detectable off-target modification at any of the tested off-target sites. Zero.
Wow. That's phenomenal. That really shows a clear pathway to achieving therapeutic level safety.
Yeah.
But I have to ask the critical question. If you need two separate guide RNAs delivered to the same cell, doesn't that present an immediate massive challenge for delivery, especially in a clinical setting?
It absolutely introduces a logistical hurdle, a big one. You need to ensure not only that both guides reach the cell, but that they are expressed at the same time and at similar levels.
And that challenge led to further refinement, didn't it? I'm thinking of the work targeting primary hyperoxaluria type 1, PH1.
Yes, exactly. PH1 is a severe, rare metabolic disorder where the liver overproduces oxalate, which leads to kidney failure. The strategy there was to knock down the glycolate oxidase, or HAO1, gene in liver cells.
And they successfully used paired Cas9 in a mouse model.
They did, delivered by AAV vectors. And to address that co-expression challenge you mentioned, they developed what they called an all-in-one system.
Meaning they packaged everything into a single virus.
Everything. Both guide RNAs and the Cas9 and protein, all into a single adeno-associated virus, or AA vector. And this optimization proved to be far superior. They achieved extremely high on-target indel frequencies, upwards of 69% in some cases, and a significant therapeutic effect.
And that's all because delivering them together guarantees they're both active in the same cell at the same time.
Precisely. It guarantees the superior co-expression you need to maximize the frequency of that required dual nicking event. So engineering the delivery vector became just as important as engineering the enzyme itself.
It really confirms that the dual nickase strategy is analogous in its specificity requirement to older systems like zinc finger nucleases and talons, which also needed two domains to work together.
Yes, but with CRISPR, it's infinitely easier to program. You're just changing the RNA sequence, not re-engineering a whole protein.
Okay, so we've moved from crude scissors to a highly specific dual-guided scalpel. But the outcome of the cut, the DSB, was still inherently problematic because it relied on the cell's unpredictable NHEJ repair. So now we need to discuss a truly radical change, engineering the enzyme to tailor the cut itself and bias the cellular repair.
This is where we shift focus from where the cut happens to how the cut actually presents to the cell. As we said, wild-type Cas9 produces a blunt cut. Both DNA strands are cleaved directly opposite each other.
And the cell sees that blunt clean break and immediately calls in the fast, error-prone NHEJ pathway.
Right. And while NHEJ is great if all you want to do is a gene knockout because an indel effectively just turns the gene off, it's disastrous if you are trying to insert a precise piece of DNA or make a single base correction.
Absolutely.
So researchers engineered specialized Cas9 variants like VCase9 specifically to alter the geometry of that cut. Instead of a blunt end, VCase9 generates a staggered cut.
What exactly does a staggered cut mean, and why does the cell respond so differently to it?
A staggered cut means that the cleavage happens at slightly different positions on the two strands, which leaves these little single-stranded overhangs. And this geometry is the key. Blunt ends are immediate substrates for the NHEJ complex. Staggered ends, however, signal the cell to engage in something called DNA resection.
Resection, meaning the cell actually chews back the DNA ends.
Yes, exactly. Enzymes come in and start to trim back those exposed single-stranded DNA ends. And that resection process actively inhibits the binding of the NHEJ machinery. So by promoting resection, you shift the repair predominance away from NHEJ and toward homologous repair pathways.
Specifically, homology-directed repair, or HDR, if you provide a donor template.
Right, and also microhomology-mediated end joining, or MMEJ.
We should pause there because MMJ is often mentioned but rarely explained simply. How does it differ from that error-prone NHEJ?
MMEJ is still a type of end joining, but it's more predictable. It uses these short, duplicated sequences, microhomologies, typically 5 to 25 base pairs long, that get exposed during that reception phase.
Okay.
So instead of just haphazardly gluing the ends, MMEJ aligns the exposed single-stranded ends using these these short homologous repeats before it sticks them together. This results in the systematic deletion of the DNA sequence between the two microhomologies, leading to a predictable outcome, a targeted deletion, rather than the semi-random indels of NHEJ.
So by engineering an enzyme like Cas9 to promote MMEJ, you actually gain a degree of control over the resulting deletion.
Yeah.
That's a very sophisticated way to manage cellular destiny.
It is, and it shows a really deep understanding of the cell's own repair biology. But of course, Cas9 is not the only option. The last decade has brought this massive expansion of the toolkit just by discovering and repurposing entirely different Cas enzymes found in nature.
Let's look at Cas12 first. It seems to solve two major logistical problems that Cas9 faced.
It does. Cas12, which you might also see called CPF1 or Cas12a, is smaller than Cas9. That's a big advantage for packaging it into AAV vectors for delivery. But more importantly, it recognizes a completely different set of targets because it favors T-rich PAM sequences, usually 5U TTTV3-all.
So it opens up entirely new regions of the genome that were previously inaccessible to Cas9 with its NG-PAM requirement.
A huge portion of the genome, yes. And mechanically, it also makes a staggered cut, generating 5' overhangs. But its truly unique feature, the one that makes it valuable beyond just genome editing, is its nonspecific collateral ssDNA transcleavage activity.
Collateral cleavage meaning once it's activated, it just starts destroying any nearby DNA that isn't its target.
Any nearby single-stranded DNA, yes. Once Cas12 successfully binds and cleaves its specific target DNA, its nuclease domain undergoes a change that activates this general nuclease activity. It just starts chewing up any nearby ssDNA molecules, completely non-specifically.
That sounds like a terrifying prospect for therapy, but we harness that for a radically different purpose. Before we get to diagnostics, what about the RNA targeting system, Cas13?
Cas13, or C2C2, represents another big departure. It targets single-stranded RNA using its HEPN domain. This means it doesn't cause any permanent changes to the host genome, which makes it ideal for temporary or tunable gene expression regulation.
So if a gene is transiently harmful or needs to be silenced for just a short period, Cas13 offers a quick, reversible solution.
A perfect one. And like Cas12, it has its own collateral cleavage ability.
But for RNA, right?
Correct. Upon binding its target RNA and getting activated, Cas13 also initiates nonspecific collateral cleavage, but it destroys any nearby ssRNA molecules.
So both Cas12, with its ssDNA collateral cleavage, and Cas13, with its ssRNA collateral cleavage, turn what sounds like a potential safety hazard into a robust detection signal. And this forms the basis of highly precise, rapid diagnostics.
It's a really elegant application of a molecular mechanism. The approach is to take a biological sample, maybe amplified RNA or DNA from a pathogen, and you combine it with the specific Cas enzyme, the guide RNA, and a special fluorescent reporter molecule.
Tell us more about that reporter molecule. How does that work?
So the reporter is crucial. It's typically a single-stranded molecule of DNA or RNA that has a fluorescent molecule on one end and a quencher molecule on the other.
And when they're close together, the quencher blocks the light from the fluorophore. The signal is dark.
Exactly. But when the Cas enzyme finds its target in the sample, its collateral activity gets switched on, and it starts indiscriminately chopping up that reporter molecule.
Which physically separates the fluorophore from the quencher.
And when they separate, the fluorescent signal becomes bright, which tells you that the target sequence is present. This is the basis of highly sensitive diagnostics like Sherlock and Detector, which are capable of detecting even single molecules of a target nucleic acid. Incredibly useful for things like rapid viral load assessments or cancer screening.
That's a powerful example of molecular repurposing. But diagnostics still relies on the cutting mechanism. We're now moving to the ultimate form of precision, which is eliminating the DSB entirely and correcting individual bases. This is the era of the search and replace function, base editing and prime editing.
This really represents a conceptual leap. Base editors, or BEs, are these remarkable modular fusion proteins. They take a Cas9 that is either nuclease-dead dCas9, which binds but doesn't cut, or a Cas9 nickase, and they fuse it directly to a deaminase enzyme.
So the Cas protein handles the high-precision targeting, but the deaminase handles the actual chemistry. It's a molecular hybrid.
It is, and the fundamental advantage over traditional Cas9 is DSB-free editing. They directly convert one base to another without inducing that unpredictable double-strand break, and without needing a separate donor template for homology-directed repair. This just massively improves the safety and predictability.
Let's start with cytosine base editors, CBEs, which convert C to T or G to A on the opposite strand. What's the mechanism behind that conversion?
The key component is acididine deaminase, which is often derived from the apobec family, a natural immune mechanism that vertebrates like us use to fight retroviruses. The guide RNA directs the complex to the site, Cas9 binds, unwinds the DNA helix, and exposes a single-stranded region we call the R loop.
Okay.
The deaminase then gets access to this exposed cytidine base and chemically converts it into uracil.
Uracil, which the cell reads as a thymine.
Exactly. Uracil is chemically almost identical to thymine, so you get a UG mismatch in the helix. When the cell repairs and replicates that DNA, it resolves that UG mismatch into a stable TA-base pair, and you've achieved your C to T transition.
That's an incredibly sophisticated manipulation of the cell's own repair mechanism. But the initial CBEs were a bit promiscuous, weren't they? They would edit multiple Cs if they were close together.
They were. The initial editing window was quite wide, maybe spanning about 5 nucleotides. So if you had neighboring cytosines, you risked editing all of them. But through really intensive engineering, researchers mutated the deaminase domains to narrow that editing window down to as little as 1 to 2 nucleotides.
The critical specificity breakthrough. It lets you discriminate between adjacent Cs and just target the pathogenic one.
It was essential. And then, of course, you have the other major transition, adenine base editors, or ABEs.
Which handle the A to G conversion.
Right. They use an adenosine deaminase that works in a similar way, converting adenine into inosine, which the cell reads as guanine, leading to an A to G base change.
Now, to maximize the efficiency of these base editors and, prevent the cell from just undoing the edit, researchers had to be clever about which Cas9 component they used and what other proteins they fused on.
Absolutely. There are 2 key modifications. First, base editors typically use a Cas9 nickase that targets the non-edited strand.
Why there?
Well, if the Cas9 nicked the strand containing that newly formed uracil, the cell might just excise that whole strand and replace it using the original unedited DNA as a template, undoing all your work. By nicking the complementary strand, you encourage the cell to use your newly edited strand as the template for repair.
That's smart. And the second modification is another fusion protein, the uracil DNA glycosylase inhibitor, or UGI. What's its role?
UGI is essential. The cell has natural defenses, like uracil DNA glycosylase, UDG, designed to find and remove uracil from the genome, because uracil is usually a sign of DNA damage.
So UDG would naturally try to erase the edit you just made.
It would. It would excise the uracil base, preventing the desired TA resolution. So by fusing UGI into the complex, you inhibit UDG. You effectively stop the cell from repeating the uracil you just created, which forces the mismatch repair system to incorporate the desired TA pair. It maximizes the efficiency of the edit and keeps indel formation below 1%.
Base editing solved single base transitions, which is a massive win. But what if the required correction is a small deletion, a small insertion, or one of the trickier transversion swaps? We needed an even more powerful search and replace tool, and that is prime editing.
Prime editing, or PE, is arguably the pinnacle of current precision engineering. It's defined as this complex fusion of the Cas9 nickase, specifically the H840A mutant, and a reverse transcriptase.
So we have the precision guidance from the Cas9 nickase, and now we have the ability to literally write new DNA via the reverse transcriptase. But the targeting element is also new, isn't it? The prime editing guide RNA, or PEG-RNA.
The PEG-RNA is the conductor of this whole orchestra. It's specialized. It contains the standard Cas9 guide sequence, plus 2 critical extensions, a primer binding site, and the desired edited DNA template sequence.
So that template is the exact piece of DNA you want to install.
Exactly, whether it's a single base change or a small insertion or deletion.
Okay, let's walk through the PE mechanism step by step. How does it work?
Okay, step 1. The Cas9 nickase nicks only one strand of the target DNA. This creates a loose, single-stranded DNA flap. Step 2. The primer binding site of the PEG-RNA hybridizes or pairs with that loose DNA flap.
It grabs onto it.
It does. And now for step 3. The reverse transcriptase, which is now positioned right at the site, uses the PEG-RNA's template sequence as a blueprint to synthesize the edited DNA sequence directly onto the target strand. It effectively writes the new sequence right into the genome.
That process creates a hybrid, right? A heteroduplex. The newly written strand is correct, but the complementary strand still has the original unedited sequence. If the cell leaves it alone, the edit could be lost. So how do systems like PE3 make sure the edit is permanent?
This is the critical resolution step. The most efficient form, PE3, introduces a second standard guide RNA that targets the unedited strand. This second guide directs the Cas9 nickase to make another nick on the unedited strand, usually many base pairs away from the original edit site.
Why the secondary nick? What does that do?
That secondary nick is a powerful signal. It forces the cellular repair machinery to prioritize repairing the break on the unedited strand. And to fix that break, the cell is strongly directed to use the newly synthesized edited strand as the template.
Ah, so it resolves the mismatch in your favor.
It guarantees the mismatch is resolved in your favor, creating a fully edited, permanent DNA duplex. If you skip that second nick, you have to rely on the cell's natural and often inefficient mismatch repair system, which frequently just excises the edit you wanted to make. It's an intentional, layered approach to guarantee the outcome.
It illustrates that continuous theme, increasing the complexity of the intervention to gain absolute control over the outcome. And just like with base editors, the components of prime editing required immediate optimization for stability.
Absolutely. The early PEG RNAs were very prone to rapid cellular degradation, which limited efficiency. So researchers developed engineered PEG RNAs, or ePEG RNAs, by incorporating these stabilizing secondary RNA structures, things like the Zika virus structural motif or G quadruplex structures.
And these additions just help the PEG RNA last longer in the cell.
That's all. And that simple change increased editing efficiency 3 to 4-fold in human cell lines.
And the innovation continues with protein fusions aimed at protecting that fragile RNA.
Yes, the PSEP editor is a great example of this. Researchers fused the entire prime editor protein complex to the LA protein, which is an endogenous human RNA binding factor.
What does La protein normally do?
It naturally protects short, functional RNAs from being degraded. So by tethering the entire prime editor complex to LA, they dramatically improved PEG RNA protection and achieved up to a 60-fold increase in precision and efficiency across multiple cell types. It just confirms that fusion is the primary driver of current innovation.
And the functional relevance of this level of precision is immense. You can finally fix complex diseases that rely on small deletions, not just point mutations.
Precisely. Prime editing has been successfully used to correct the CFTR F808del mutation, which is the most common cause of cystic fibrosis, and they did this in patient-derived organoid models. The ability to install a small, precise replacement sequence, not just a base swap, truly changes the therapeutic landscape. We've transitioned from the blunt tool of NHJ to the ability to write new information directly into the genome.
Okay, now let's shift gears completely. How has this molecular renaissance translated into real-world applications across different sectors, from agriculture to clinical trials?
Well, the ability to program the genome so easily has just exploded across basic science, agriculture, and medicine. In agriculture, the global impact is immediate, particularly in countries like Japan, where CRISPR-edited food has already entered the commercial market.
What specific types of agricultural products are reaching consumers right now because of this editing?
The focus is really on enhancing nutritional profiles and improving yield efficiency. For example, Japan is selling GABA-rich tomatoes. CRISPR editing was used to enhance the production of the neurotransmitter GABA, which potentially offers calming effects. And in aquaculture, they've successfully modified sea bream and pufferfish.
What do they do to the fish?
In these fish, they used Cas9 to disrupt the leptin gene, which is what regulates appetite.
So by disrupting leptin, they essentially tell the fish that it's constantly hungry.
That's right. And this modification allows the fish to eat more and grow faster. One edited species was shown to reach twice the size of natural specimens, and another, where they disabled the myostatin gene to promote muscle growth, showed a significant increase in muscle mass. It's all about maximizing food production efficiency.
And for essential crops, we've seen attempts to address global food security issues like yield and disease resistance.
Yes, for staple crops like rice and wheat, strategies involve increasing yields by knocking out the CKX enzyme, which degrades the plant hormone cytokinin. More cytokinin means higher yields.
Yeah.
Other examples include engineering resistance to devastating diseases like fungal infections in wheat and various viruses in rice, and modifying quality things like creating low-gluten wheat varieties and high oleic acid soybeans. It's just rapid, targeted breeding without introducing any foreign genes.
Beyond food production, the precision offered by base and prime editing is really essential for creating high-fidelity research models. Moving past simple gene knockouts is critical for studying complex human diseases.
This is a major area of impact. Animal models often fail to replicate human disease accurately. Prime editing allows researchers to precisely introduce the exact point mutations found in human patients into animal models like mice and rabbits.
Can you give us a concrete example of a model that benefits from this extreme precision?
Certainly. We've seen the creation of models for severe genetic disorders like Tay-Sachs disease, where a specific mutation in the HEXA gene was introduced into rabbits. More subtly, in mice, researchers have used prime editing to precisely introduce mutations in the HOX13 gene, which is essential for limb development, and in the X-linked androgen receptor.
So these aren't simple knockouts. They are accurate representations of human pathogenic point mutations.
Exactly, which allows researchers to study disease progression in a manner that was previously impossible.
Let's turn our attention now to the most visible area: clinical advances in human therapy. Cancer immunotherapy, specifically engineering a patient's own immune cells, seems to be dominating the phase 1 and 2 clinical trials.
It is. Cancer immunotherapy relies on adoptive cell transfer, or ACT, where you harvest T cells, modify them, expand them, and reinfuse them. Most of these trials are using Cas9 ribonucleoprotein or RNP complexes ex vivo to enhance these T cells' ability to fight tumors.
Whether they are CRT cells or TCR-T cells.
Right. And the key genetic disruptions they're implementing are primarily to genes that act as cellular brakes. For example, knocking out the PD-1 gene, which is an inhibitory checkpoint on the T cell, thereby boosting its anti-tumor immunity.
And in allogeneic therapies, where the T cells come from a donor.
There, they disrupt the TRAC, which is the T cell receptor alpha chain. This prevents the cells from recognizing the host as foreign, which prevents the potentially fatal graft versus host disease.
And in the treatment of solid tumors, I understand they were getting even more aggressive with multiplex editing, knocking out multiple genes at the same time.
They have to, because the tumor microenvironment is so highly suppressive. Clinical strategies now involve simultaneously knocking out targets like CD70, B2M, Regnase-1, and TGFR2, all of which are aimed at improving the T cell's persistence, its ability to get into the tumor, and its resistance to those harsh suppressive signals.
And the early data looks good.
It's encouraging. It's showing that this RNP-mediated gene disruption is relatively safe, with no detectable off-target effects in the edited cells being reinfused.
Beyond cancer, the success story of CRISPR-Cas9 in genetic blood disorders is now a clinical reality, culminating in a major regulatory approval.
This is the landmark achievement for sickle cell disease and beta thalassemia. And the strategy isn't to correct the point mutation causing the disease, but rather to reactivate a fetal developmental program.
How do they do that?
They edit the patient's hematopoietic stem cells ex vivo using Cas9 to disrupt the BCL11A erythroid-specific enhancer.
And what happens when they disrupt that BCL11A enhancer?
So BCL11A normally acts as a silencer. It turns off the production of fetal hemoglobin after birth. By disrupting the enhancer that drives BCL11A expression specifically in red blood cells, they essentially keep that fetal hemoglobin pathway turned on.
And fetal hemoglobin is unaffected by the sickle cell mutation.
Exactly. It effectively reverses the disease symptoms. This success led to the FDA approval of Casgevy, which marks the first cell-based gene therapy using CRISPR-Cas9 technology. That approval signaled to the entire medical community that the safety and efficacy concerns of CRISPR could, in fact, be managed.
And we've also achieved a critical milestone for in vivo editing, where the components are injected directly into the patient's body.
Yes, the first successful in vivo editing milestone was the treatment of hereditary transthyretin amyloidosis, or ATTR, back in 2021. This is a life-threatening disease where the liver produces a misfolded TTR protein that accumulates in tissues.
And patients just received a single intravenous injection.
A single 5e injection of an LNP-encapsulated CRISPR-Cas9 system. The system targeted the liver cells, resulting in a permanent modification that significantly knocked down the production of that TTR protein. This one intervention resulted in substantial clinical benefits, and it highlighted that in vivo delivery to certain organs is achievable and highly effective.
Let's connect back to base and prime editing. We see the success of Cas9, but now we have a major clinical milestone for prime editing itself.
This is the future unfolding right now. Prime Medicine's therapeutic candidate, PM359, is the first prime editing therapeutic to enter a clinical trial. It's targeting chronic granulomatous disease, or CGD, which is a rare immunodeficiency.
But what's the specific mutation being targeted in CGD?
CGD is caused by mutations in the NCF1 gene, which leads to a deficiency in the NADPH oxidase enzyme complex. This is critical for immune cells to fight infections. So PM359 is editing hematopoietic stem cells ex vivo to correct this precise mutation.
And the initial results?
The initial data released showed safety and, crucially, efficacy. It demonstrated that a single dose restored that NADPH oxidase activity in the patient's edited immune cells. This confirms the feasibility, the safety, and the functional utility of this incredibly complex prime editing machinery in humans.
All this precision, the nickases, base editors, prime editors, it's all phenomenal, but it's completely moot if we can't get the tools safely and efficiently inside the correct cell. Delivery remains the persistent challenge that really dictates the entire field's clinical viability.
Delivery is the critical bottleneck. It truly is. The choice of cargo and the choice of vehicle profoundly influences both the efficiency and the safety profile. We typically classify cargo into three types.
Let's look at those cargo types and their risks.
First, you have DNA plasmids, which encode the Cas enzyme and the guide RNA. They're easy to produce, but they result in sustained gene expression because the cell just keeps reading the DNA. This sustained activity translates to the highest risk of off-target effects.
Okay, what's next?
Second is Cas9 mRNA plus the guide RNA. This is better because the activity is transient. The mRNA degrades over time. But the safest and increasingly the gold standard for therapeutics is the ribonucleoprotein complex, or RNP.
The RNP is just the Cas protein bound directly to the gRNA. Why is that the gold standard for safety?
Because RNP offers highly transient activity. The protein complex is active immediately when it enters the cell, but it's degraded by the cell within hours. This hit-and-run strategy minimizes the time the Cas enzyme is active, which leads to the lowest possible off-target risk, and that's absolutely essential for any therapeutic application.
Okay, now what about the vehicles that transport these cargoes into the cells? We generally rely on viral and non-viral methods.
Viral vectors, particularly adeno-associated virus, AAV, and lentivirus, LV, are the most common in clinical trials just because of their high efficacy. However, AAV faces severe limitations.
Well, like size capacity.
Exactly. Some of the newer, larger prime editors are a real struggle to package into an AAV, and AAV also carries a risk of immunogenicity, as many patients have pre-existing antibodies against the viral capsid.
And lentivirus.
Lentiviruses are excellent for ex vivo modification, like T cells, but they carry the inherent risk of integrating the cargo into the host genome. While integration-deficient LVs have been developed, the general safety profile is still under intense scrutiny. This is really pushing the field toward non-viral solutions.
And that brings us to the lipid-based systems, which are rapidly evolving and solving many of the problems viral vectors present.
Non-viral methods like lipid nanoparticles, LNPs, and liposomes are gaining immense favor. They're versatile, scalable, generally safer, and far less immunogenic than AAVs. Crucially, they have a larger capacity, which makes them suitable for packaging the massive fusion proteins required for prime editing.
And the major innovation in LNP delivery that connects directly back to our theme of precision is selective organ targeting.
That's right. LNPs are just these little lipid shells, and their surface chemistry can be engineered. Researchers can decorate LNPs with targeting moieties, like specific peptides or aptamers that bind selectively to receptors found predominantly on the surface of cells in, say, the liver or the spleen or the lungs.
So instead of just injecting the LNPs and hoping they diffuse to the right tissue, you engineer the surface to actively seek out and bind only to the target cell type.
Exactly. This drastically increases the local concentration of the CRISPR components at the intended site while minimizing exposure and potential toxicity in non-target tissues. We're seeing this merger of delivery science and molecular engineering where the vector itself becomes a tool for precision.
What a journey. From a bacterial defense mechanism that was, initially too sloppy to use safely, to FDA-approved therapeutics and highly sophisticated tools capable of writing new genetic information into the human genome with astonishing precision.
It's truly remarkable. To synthesize this whole evolution, the core narrative is the systematic elimination of cellular unpredictability. We started with wild-type Cas9, creating these blunt DSBs repaired haphazardly by NHEJ. We solved the binding specificity problem with nickases and high-fidelity Cas9 variants.
Right.
Then we solved the repair outcome problem by engineering nucleases like vCas9 and Cas12 to favor the more predictable MMEJ and HDR via staggered cuts. And finally, we just circumvented the entire issue of DSBs with base editors and prime editors.
Moving to DSB-free, programmable base swaps and small insertions and deletions.
Exactly, which dramatically enhance the therapeutic safety profile.
That progression from blunt cut to dual nick to chemical conversion is the very definition of molecular engineering. It's no longer about what the enzyme is, but what we can make it do through fusion.
That's the key takeaway about all the current innovation. The creation of base editors and prime editors fundamentally relies on modular fusion, marrying Cas9's incredible targeting ability with powerful existing enzymes from other domains. Base editors fuse Cas9 with deaminases, which, as we discussed, were repurposed from our own vertebrate immune systems.
Like ApoBec.
Right. Prime editors fuse Cas9 nickase to reverse transcriptases. We're even seeing these complex systems like pasty and passage fusing Cas9 to huge enzymes like serine recombinases and transposases to achieve highly efficient large-scale gene integration.
So the scientific imperative now isn't just about finding a better Cas enzyme, but about finding the next perfect enzyme in nature to attach to the Cas targeting domain.
Precisely. And that raises the final provocative thought for you to consider. The foundational targeting mechanism of this entire system relies on that 20 nucleotide guide sequence and the required PAM sequence. We have successfully solved so many problems by repurposing and fusing different enzymes to Cas. So, given the success, what other natural enzymes beyond the current nucleases, deaminases, and reverse transcriptases will scientists adapt and fuse with the CRISPR targeting capability to solve the remaining fundamental challenges, like the strict PAM requirement, or to create entirely new types of precise genomic or epigenomic modifications we can't even imagine today?
That question really highlights the modularity, doesn't it? The future of genomic editing might not be about finding the next Cas protein at all, but about constantly finding and bolting on new, highly specialized tools. What is the next ApoBec, the next reverse transcriptase, waiting to be discovered and repurposed for the genome?
Indeed, it's a question for the next generation of precision engineers.
That's a wrap on this deep dive into the rapid evolution of CRISPR precision. Thank you for guiding us through this molecular renaissance.
My pleasure.
We'll see you next time on the Deep Dive.